frameshift mutation stopped by the terminator. What if the frameshift mutation didn't happen up here? What if it happened down here? Okay, so maybe I just screwed up the last codon in the DNA, in the gene, right? But if if the frameshift mutation is in the beginning of the gene, then you're going to screw up the whole gene. But either way, the terminator stops it. It stays, the frameshift mutation stays in the gene. But what if the gene were a gene for an essential enzyme? An enzyme to run your Krebs cycle, perhaps. An enzyme to run your glycolysis, perhaps. Then it could be fatal to the organism, or the cell, whichever way you want to think of it, because with bacteria, the cell is the organism, right? And that's why they study operon theory in bacteria, because they're a lot simpler organisms. Okay, so you understand the different types of mutations, and the degeneracy of the genetic code, and what a mutagen is, and now we're going to do this over here. And then we'll do induction and repression. Okay. Now, protein synthesis steps. This is the one thing I didn't do, was do the steps with you. I kind of went through the process without telling you the names of the steps. So let's put a big bacterial cell up here. Plasma membrane, cell wall, nucleoid. Let's do our flower-powered nucleoid. We're going to open up part of our nucleoid because we're going to make mRNA. So this is your double-stranded DNA, right? You've got your little hydrogen bonds. Not very good at drawing my dotted lines. Okay. The hydrogen bonds temporarily break. mRNA is copied from one side of the DNA. Here would be your promoter region and your terminator region. On your mRNA, there would be a start and a stop. And the functional gene codons would be in the middle, if that helps at all. When the mRNA is done being copied, it literally pops out, leaves the nucleoid, and goes out to the cytoplasm, where it's translated on a ribosome. And as it's translated on a ribosome, you end up getting a chain of amino acids, right? As your mRNA is read, you get a chain of amino acids, and it twists and bends and folds into a functional protein, okay? The first step, protein synthesis steps, actually there's an initiation step. I won't ask you about initiation. Just means when it's being copied, the mRNA is being copied at the DNA, it has an area that says, okay, let's go. Let's start the process of protein synthesis. It's a signal to the cell. First step is transcription. Transcription happens at the nucleoid. Cell makes a copy, an mRNA copy, of the DNA for one gene, the gene that codes for the needed protein. Second step, translation. In translation, the mRNA goes to a ribosome in the cytoplasm. That's where that's going to happen. So the ribosome is the rRNA. So the mRNA goes to rRNA and is translated by tRNA into a chain of amino acids. Then the last part that I generally don't ask you about is termination. Termination is when the tRNAs get to the stop codon. They know they're done making the protein. The protein is cleaved off of the ribosome, and in the cytoplasm, it might finish assuming its shape. Protein takes shape, let's say. It takes shape as it's being translated, and then it leaves the ribosome as a functional protein. Because its shape determines its function. Its shape means it's got certain shape active sites that'll bind with a particular substrate, and the enzyme will be able to run its reaction to convert the substrate into something.
Okay, so what's this? Cell makes an mRNA copy of DNA for what exactly is this letter for?
Cell makes an mRNA copy of the DNA for one gene.
Okay.
For one gene. The gene that codes for the needed protein. Let's do shopping list over here.